you go. So our session today is being delivered by Professor Bruce Lambert, who's calling us from Chicago, and will be giving us a seminar entitled Psycholinguistic Tests Predict Real-World Drug Name Confusion Error Rates. Bruce received his PhD in speech communication from the University of Illinois. He's professor in the Department of Communication Studies in the Department of Medical Social Sciences at Northwestern University, and an adjunct professor in the Department of Pharmacy Systems, Outcomes and Policy at the University of Illinois in Chicago. His research focuses on health communication, clinical informatics, patient and medication safety, and medical liability reform. He's also president of BLL Consulting, a firm that solves problems involving health communication and technology. He's got a huge amount of experience in this topic, and we'll be hearing about a small part of it today. And this, I won't give you too much about the seminar detail, but this is about wrong drug medication errors, which are common, and Bruce will be talking about two particular papers during today's presentation. So just to let you know again, the session is being recorded and will be available online. We'll share a link. If you have questions during Bruce's seminar, please do put them in the chat, and there should be time at the end for us to come back and pick those up. I think that's about it. So I'll ask Bruce to share his slides and let Bruce get on with his talk.
Good morning, everybody. And thanks for having me here today. I look forward to talking to you about this work that I've done on trying to prevent confusing drug names from entering the marketplace. I've been studying drug name confusions for a very long time, 31 years or so, and I joke sometimes that I haven't solved it yet. You know, unfortunately, today in hospitals and health systems around the world, drug name confusions will still occur. But I think we made some progress, and I want to talk about two particular papers where I showed that laboratory tests of drug name confusion are significantly associated with real-world error rates in large pharmacy systems. So that's what I want to talk about. There's two papers that I've published on this topic 8 years apart. Most of this work took place a relatively long time ago, you know, in 2015, 16, 17. But the second paper got delayed and came out just last year. And that's the one that caused Will to reach out and ask if I would talk about it. So that's what I'm going to talk about today. I'm going to give you the context of both of these studies so you can see what we did, because they're on essentially the same data set and approaching them in just two slightly different ways for cross-validation. So this is the topic here. The patient safety topic is wrong drug errors, particularly look-alike and sound-alike medication errors. It's stunning what we found over the years, not just us, but lots of other people who have looked at this. If you review the literature on wrong drug errors, as W. Edwards Deming would say, the system is perfectly designed to produce about 1 in 1,000 wrong drug errors. So if you look at in the hospital, if you look at medication orders, about 1 in 1,000 of those are for the wrong drug. And if you look in pharmacy systems, about 1 in 1,000 medications dispensed are for the wrong drug. I mean, it's absolutely astounding, the consistency of this rate. So you think on the one hand, well, that means that 99.9% of the time we get patients the right drug. So well, our work is done here. We're doing great. Unfortunately, you know, in the US, there's 4 billion prescriptions dispensed in the outpatient setting every year, which means 1 in 1,000 errors results in 4 million wrong drug errors per year. And you could scale those numbers to the UK. They would be a little bit lower, but on a proportional basis, they're likely to be about exactly the same. So we'd like to do better than this. We wouldn't like it, you know, in lots of other domains if we were getting things wrong 1 out of 1,000 times. And for those of you on this call, you know probably all about drug name confusion errors. I just listed some typical examples, you know, hydroxyzine, hydralazine, of which you can find case reports of hydroxyzine, hydralazine in the early 1970s when I first started getting into this. In fact, you can find ancients. I think I have a quote from Galen talking about how drug names can be confused. So this is a very, very old problem. But these are modern examples: Brilinta and Brintellix and clozapine and clopidogrel. And there are hundreds of other notoriously confusing pairs. And so I want to talk today about how we might prevent these in the upstream fashion. That is, how do we prevent confusing names from getting on the market in the first place, which is to give regulators and drug companies better tools for identifying what might turn out to be confusing names. So there are lots of pre-approval screening methods already in place in the US at the FDA and the EMEA and elsewhere, where they use expert panels, they use computerized searches, but most of these methods lack any empirical validation. We don't really know whether they work. And if you look at the fact that, confusing names continue to enter the marketplace, there's an eye drop, and there's this example of Duracell and Durazol. One was an eye drop and one was a wart remover. And somehow one name got on the market when the other was already on the market, even though they were obviously extremely similar. So we wanted to see if we could validate some of these pre-approval screening methods against real-world error data, such that we might be able to encourage the regulators to use these methods more, and the companies too. Because sometimes if you ask the companies to do more pre-approval screening, they say, oh, these are not validated measures. Why should we have to do this? They complain that they're over-regulated and so on. So we tried to counter this objection by producing empirical validation. And despite, decades of trying to solve this problem, these drug name confusion errors remain stubbornly prevalent. In spite of my spending my entire career on this problem, drug name confusions continue to happen and continue to harm patients, and occasionally to kill patients. There's some notorious series of case studies with cisplatin and carboplatin, where people were getting cisplatin at carboplatin doses and always dying, and dying really the most horrible death you can imagine, because they would get cisplatin at a carboplatin dose and there's nothing you could do once they got this overdose. It was uniformly fatal, and there's a bunch of those. So we'd like to stop that from happening if we could. So here's just more examples, the antidepressants and blood thinners getting mixed up, more than 50 FDA reports, which led to a name change. This is Duracell and Durazol. One was a wart remover, one was an eye drop. And of course, someone put the wart remover in their eye and blinded themselves in one eye, unfortunately, in Navane and Norvasc. And there are many, many, many others. And you can look at the FDA, ISMP, and other regulatory organizations have published long lists of confusing pairs of drug names. So our hypothesis here is that drug names confusions are not random. They're rooted in the same basic psychological mechanism, and that is that human beings are vulnerable to similarity-based confusions. And in a sense, this means we will never completely eradicate these errors, because human beings are simply vulnerable to visual similarity, auditory similarity. Because of the way memory and perception work in human beings, similarity will always trip us up at some rate. So we think, because there's a fundamental cognitive mechanism, if we can replicate these failures in the laboratory, we should be able to predict the relative confusability of real-world name confusions as well. So we had 3 different kinds of tasks that we had participants do in these experiments. One where a visual presentation, where we briefly present a drug name, we mask it by visual noise, and we have them try to identify the name they saw. Then we had an auditory perception one where they put on headphones. We played the name against a background of 20-speaker babble, like trying to hear a name in a cocktail party or in a busy pharmacy or medical ward. And then there was a short-term memory experiment where we showed them a drug name. Then we had them do a mental arithmetic task. And then we asked them what drug name had they seen before. And that's meant to mimic short-term memory interference that you might have when you see or hear a drug name and then get interrupted and then have to try to remember the drug name that you saw or heard. And the main question is, of course, do the error rates we observe in these laboratory tests predict the error rates that we see in the real world? And in this case, the real world is a set of self-reported errors we got from a large pharmacy system in the US, a pharmacy system that had more than 1,000 sites in the US, which shall go unnamed. It's a patient safety organization, a sort of official patient safety organization associated with one of the large chain pharmacies in the US. So I'm going to talk about 2 different studies, one which we published in 2017, one which we published in 2025. And I want to talk about the differences between them. In the first study, we were looking at pairs of names. We wanted to know, could we predict the error rate for a specific pair, for something like hydroxyzine and hydralazine. So in the experiments, we would show people hydroxyzine and hydralazine, and we would see how often, for example, we would show them hydroxyzine, and then we would show them hydroxyzine and hydralazine and pick them and say, which one did you just see? And And so the unit of analysis in the 2017 study was specific pairs of names, because this has often been the focus in the literature, because ISMP and others, they publish these pairs, FDA too. And so we looked at 107 pairs of drug names. We did these 4 psycholinguistic tests, and we said, do the tests predict the confusion in the real world? And they did. And in 2025, we said, let's not focus on the pair as the unit of analysis, but on the individual name, because hydroxyzine is not only confused with hydralazine, but might be confused with hydrochlorothiazide or other names. In fact, in this data set we have, the individual names were often confused with 2, 3, 4, 5 other names. So it's not just a question of pairs. So the second study is about with the individual name and all of its error partners, that might be confused. And we wanted to see, would our psycholinguistic tests predict the error rate at the level of the individual name? And again, it did. So that's the foreshadowing. So this is just a schematic diagram of the design of these studies. So the first is what we call backward masking, meaning that the visual presentation of the name was backwardly masked by this row of X's. So they would fixate on this plus sign on the screen. These were all done on a little MacBook. And the participants were pharmacists, nurses, physicians, and pharmacy technicians in the first study. And in the second study, just pharmacists and pharmacy technicians. So they fixate on this plus sign. Then we would show them a name like lorazepam. Then we would mask backwardly the name with this row of X's. And then they would have to type the name in a free response. And then there would be a drop-down menu where we would ask them to select the name from a list of names as well. And then there was this speech and noise test. They would fixate on a plus sign. They'd be wearing headphones. They would hear someone say the word lorazepam against a background of 20 speaker babble. Then they would type the name in a free response and then select the name from a drop-down menu. And the short-term memory test as well. So there's lorazepam and then a mental arithmetic problem, and then they would type the name or pick from a drop-down menu. So all of these were meant to mimic real-world perception tasks that pharmacists, nurses, physicians, and technicians have to face under different circumstances of degradation or cognitive load. And one of the reasons we do degradation and cognitive, there are 2 reasons that we do degradation and cognitive load. One is because there's a lot of degradation and cognitive load in the real world, and we're meant to mimic that. And the second is that in the experiments, the error rate, if we didn't do this degradation, let's speeded presentation and so on, and noise, the error rate would be so low we wouldn't be able to measure it. So this is a way to artificially inflate the error rate for the experiment so we could measure it. And it's standard in psycholinguistic experimentation. This is just another schematic representation of the tasks. And this is a schematic representation of the difference between the study we did in 2017 and the study we did in 2025. So again, in 2017, the scope was just specific pairs like hydroxyzine, hydralazine. What's the error rate between those names? That's what we were trying to predict across 105 pairs or whatever it was. But in reality, first of all, regulators don't approve pairs of names. Regulators are confronted with the choice of an individual new name. Is this new name that's proposed to go on the market confusable or not? And so we tried to, in this more recent study, focus on that question. So you have a new drug name, and it's potentially confusable with lots of what we call competitors, with lots of similar existing names. And so what we tried to do in this more recent study is predict an individual drug name's sum total error rate across all of its competitors. So again, hydroxyzine is not just confused with hydralazine, but is also confused with hydrochlorothiazide and all sorts of other names. So this is just the summary of what we did in the 2017 study, 80 participants. Oh, there were lay people in the 2017 study. We found that there weren't very big differences between doctors, nurses, pharmacists, and technicians. So that's why in the 2025 study, we just dropped the doctors and nurses and did pharmacists and technicians. It's expensive to recruit doctors in the US. And so it was easier for us to get pharmacists and technicians. We still paid people $100 to do these trials because it takes about an hour because there's hundreds of drug names that they go through and 3 different tasks. So this is a summary of what we found in 2017. So this is on the vertical axis, the observed error rate. So let me tell you a little bit about the data again. So we have this a pharmacy system that's in the US called a patient safety organization. This is in the US, you can sign up to be a patient safety organization that gives you some legal protection when you report error rates, and you can report them to a kind of national system of error reporting. So there are many of these patient safety organizations in the US. There's a procedure for signing up to become one. And this particular large pharmacy system was a patient safety organization. I had a relationship with them and they agreed. It was not easy, believe it or not, to get the pharmacy chain to send me all the detailed error data. So this is this data set of every self-reported wrong drug error in this pharmacy system in a given year. So this pharmacy system dispensed millions and millions of prescriptions over the year, and for every name that there was at least one self-reported wrong drug error, I would get the name that was intended to be dispensed, let's say hydroxyzine, and then the name that was the drug that was actually dispensed, let's say hydralazine. And we would get that for every instance of a self-reported error. So we can acknowledge off the bat that self-reported errors are far from perfect. We know that self-reported errors suffer from lots of biases, the most obvious bias being under-reporting. We know from all sorts of other studies that if you compare various rates of measuring error reports, they all capture different errors. If you compare chart review to direct observation to self-report to patient report, you don't get the same rate and you don't even get the same errors. Chart review discovers different errors than direct observation, which discovers different errors than self-report, which discovers different errors than patient or family report. But this particular study is all these self-report from the chains. Which are not perfect, but you would think to be a reasonable indicator of the true error rate. And then on the horizontal axis, we have predicted error rate that's in the laboratory, and the vertical axis, the observed error rate. So it's not a perfect correlation, right? The regression model explained about 37% of the variability. And you could say it's not perfectly linear. We could have modeled this as a curvilinear model, but linear was satisfactory. And then what we did in the first experiment in 2017, we actually had two separate data sets from this patient safety organization. And we built the model on sort of data set one, where the R squared was 0.37. And then as validation, without changing the weights in the model at all, we froze the weights in the regression model and then applied it to 77 overlapping pairs from data set two, where we had data on error pairs from a different data set. And the R squared there was 0.45. So this is strong cross-validation across systems in this patient safety data set. So what this shows is that laboratory-based measures of confusability at the level of the individual pair are significantly associated with measures of error rates in the real world, at least as far as these self-reported errors are from a large pharmacy chain. And this is just a summary of some of the cross-validation we did in 2017. We did two levels of cross-validation in 2017. The first is an internal cross-validation, what's called tenfold or n-fold cross-validation, which is you take the one data set, you train on 90% of the pairs. That's like you build a regression model on 90% of the pairs. Then you test the model on the held out 10% of the pairs, and you repeat that 10 times and you average the error rate. And because the concern is when you build the full model, it's like you're training on the test set. So there's good reason to believe you could be overfitting to the data. But when we did this cross-validation, the error rate on the cross-validated set was only 1.8% higher than the error rate on the full data set, meaning we were not overfitting by very much. We were capturing the real structure of the underlying data. Had we been overfitting, like training on the test set to an extreme extent, then we wouldn't have been able to do so well on cross-validation. Cross-validation was only a tiny bit worse than the whole data set. And then we did this other kind of external cross-validation, where we had these 64 pairs from this second data set were present in the first data set. So we froze the weights on the first model, applied them to the second model, and the R squared there, the fit was even stronger. It was 45%. So what this shows us is that there's something in this regression model, which is primarily a function of the error rate. It's mostly the main predictor is the error rate in the laboratory tests, that this is a predictor even in the external cross-validation data set, the second pharmacy chain, from which we had not seen any data when we trained the regression model. So going to the study that we did in 2025, here we had 37 participants, about half pharmacists, half pharmacy technicians. They were relatively young people. And we had them at the end of the experiment. They filled out a simple measure of their familiarity with the underlying drug names. And not surprisingly, pharmacists and pharmacy technicians are on average extremely familiar with the drug names that we showed them, because these were names that were relatively commonly prescribed in community pharmacy, or commonly seen in community pharmacy practice. So this is a real-world data source. This is an AHRQ listed patient safety organization. So AHRQ in the United States is the Agency for Healthcare Research and Quality. And these patient safety organizations are set up through this federal agency. We had 18 months of self-reported errors from more than 1,000 pharmacy locations from a large U.S. retail pharmacy chain. And in this particular study, we picked 77 individual names to analyze. The real-world error rates were highly variable, ranging from 1 per million prescriptions to 2,000 per million prescriptions. And this is the same pharmacy chain as in the 2017 external validation. But again, we're looking at individual names, not pairs in this study. So that's the background. And what do we find? We remember we gave them 3 different cognitive tests here. We gave them backward masking, that's the visual perception one, speech and noise, and short-term memory. And another subtlety of these papers, that there were 2 different responses that every participant made to every trial of the experiment. The first response was free response. So we show them hydroxyzine for just a fraction of a second. Then we take it away, we mask it by these X's. And then we say, what did you just see? And they have to type in what they saw. That's free response. And because there's some misspellings and typographical errors, we gave them credit for a correct response if the free response answer was within an edit distance of 1 character, meaning if what they typed in could have been transformed into the correct spelling by an insertion, deletion, or substitution of just 1 letter. So we've done this in the past, just to correct for the possibility of a spelling error or a typographical error, even though they really did identify the correct name. So what I'm showing here are the free recall error rates, which is the error rate based on this free response, not the drop-down menu option. Those were significantly associated with real-world errors as well, but I'm just showing you these in this particular slide. So the error rate in visual perception was 56%. And remember, the name was presented for only 48 milliseconds. This is barely like the blink of an eye. In fact, when participants first do the experiment, when they do practice trials, they often laugh because the name appears so quickly that they think, well, this is an impossible task. And sure enough, they get it wrong almost 60% of the time. But the interesting thing is 40% of the time, even when the name is presented for only just a fraction of a second, they get it right. That's because visual perception is just astoundingly accurate in human beings, especially for familiar stimuli. And auditory perception, the error rate was 38%. And this is against this background of like cocktail party noise. And short-term memory, that was the easiest task for them. The recall error rate was only 24%. This is when they had to do the arithmetic problem without forgetting. And I say here at the bottom, each test is an imperfect indicator of a shared latent characteristic of the drug names, which you could just think of as their inherent confusability. And I've published a lot of other papers trying to get at what is this inherent confusability really about. You could just think of every drug name as existing in the neighborhood of other similar drug names. And every drug name, all the names in the neighborhood have an underlying familiarity, which is really a function of prescribing frequency. So if you have a relatively rarely prescribed name in the neighborhood of lots frequently prescribed names, it's very likely to be confused. The frequency of prescribing is like the mass of a planet, which draws attention towards it by gravity. So if you have the most dangerous situation is a relatively infrequently prescribed drug in the close neighborhood of a frequently prescribed drug. But I don't deal with all that stuff here, but that's just the mechanism by which this happens, is that we think the underlying confusability of a name is due to its frequency weighted neighborhood similarity, which is how many confusing names are in its near neighborhood and how commonly prescribed are those near neighbors. So what do we find here? I give the individual correlations between the error rate and free recall and forced choice and the real world error rate for these individual 77 names. And we take the log of the real world error rate in order to improve the statistical fit of these models and to deal better with some of the assumptions of the models. But it's relatively common to take the log of the error rates to make them behave better. So as you see, the correlations are relatively high across all of the tasks. But there's an interesting thing happens here that sort of terms of signal detection and noise reduction. If you take the average of all the tests across both response types, the correlation is higher than it is for any given task. So backward masking free recall, the correlation is 0.47, and short-term memory free recall is 0.36. But if you average all of them together and use that mean to predict the real-world error rate, the correlation is 0.5. So what do we conclude, at least from this correlational analysis? Laboratory error rates are significantly correlated with real-world error rates at the level of the individual name, just as they were at the level of the pair of names. Now, in a way, you could say we should have published this paper in the Journal of Obvious Results because the similarity predicts confusability. Anyone who knows anything about how word recognition works in human beings in the visual and auditory system knows that if you increase similarity, you will increase confusability. The way that word recognition is organized neurologically, cognitively, is such that we are very, very sensitive to when we see a particular name, it activates a set of names in our mind, and then we select the name essentially that has the highest activation. But similar names, so when you see hydroxyzine, it loosely activates hydralazine as well, and that's what promotes the confusability. So this is a strong effect. Correlations of 0.5 in this kind of behavioral research, especially when you have a real world outcome, is pretty high. So what this shows you is that if you're a regulator, you can be confident this is a validated test. Drug names that fail these laboratory tests, that have high error rates in the lab, are significantly more likely to be involved in real world dispensing errors. Now, there's lots of other errors we didn't study. You could say, well, what about administration errors? We have another study ongoing here at Northwestern hospital system, which is an 11 hospital system around Chicago, where we're looking at the relationship between tall man lettering and barcode warning rates. But there, the unit of analysis is nursing administration errors. Here in these papers, it's all pharmacy dispensing errors. So you could say, well, this is great. It predicts pharmacy dispensing errors. But what about order entry errors in CPOE? What about nursing administration errors? What about patient self-admit? We didn't measure any of those things. So the argument, you could say we don't know for sure, but the argument is there's a fundamental cognitive process that we're tapping into here, and that is the visual and auditory sensitivity to similarity. So our strong prediction would be that these tests would predict nursing administration error rates, patient self-administration error rates, and physician CPOE order entry rates. But I don't have that data directly, so we just have to say we don't know for sure. We just have a strong suspicion. This is when we put this into a regression model with some other factors that we thought might be related. So the reaction time to a correct response. So at first, the top line, the laboratory error rate, that's the main predictor. And sure enough, it was the strongest predictor. The laboratory error rate was strongly and significantly predicted with, or associated with the real world error rate. Reaction time, which is how long did it take you to arrive at a correct response, was negatively associated, which is what you would expect. The longer it takes, the more confusable the name is, because you're trying to distinguish between two confusing names in memory. And then other characteristics of the name, the bigram frequency, there's how many two-letter subsequences did the two names have in common, subjective familiarity, and even just the length of the name. These things were not significantly associated with the error rate in the real world. So these are relatively good fitting models. R squared of 0.35, again, which is in this kind of behavioral research with a real world outcome, is pretty strong. And even when you take out the laboratory error rate, The R squared of the model is 0.17. When you add it back in, it becomes 0.35. So the lab error rate is adding a significant amount to the other predictors. This is just more validation that the laboratory error rate is the strongest predictor, and each unit increase in the lab measured error rate is associated with a 9.2 unit increase in the log real world error rate. Now, those terms are a little hard to deal with intuitively. The most important thing to understand is just that it's a strong and significant predictor of the real world error rate. So this is just a summary of the regression model. The R squared is 0.35. So the model explains 35% of the variability in real world errors. The primary driver is experimental error rate, and adding lab error rate to the other models improves prediction very significantly. It's the main driver of the predictive power of the model, in addition to reaction time. So this is a graph that we saw from 2017. This is the graph from 2025. Again, on the vertical axis is the log transformed observed real world error rate. On the horizontal axis is the predicted error rate, which is the laboratory error rate. And again, you see here is this fit is more linear than the other one, which is slightly curvilinear. And I summarized some of the error rates as well. This is just a visual depiction of the association we were talking about before. We then did another thing. So in this study, there were only 77 observations because there was one rate for each name, and there were only 77 names. But the thing is, there were more than 11,000 trials across these participants. So when you take, but for every name, you had to take the mean error rate. So you lose a lot of observations by doing that. So I said, well, I don't want to lose all that power. Let's do it the other way around. So here we flipped everything around. Instead of using laboratory error rate to predict the real world error rate, where laboratory error rate had to be a mean, we flipped it around and said, let's use real world error rate to predict whether or not on a given trial the participant made a mistake. And we had 11,088 individual trials. So an individual trial is when a participant sees a name. And those trials are binary. They either get it right or they get it wrong. So this is a logistic model of 11,000 trials versus only 77 observations in the previous model. So, and it worked this way as well. So what this shows is that the real world error rate predicts the laboratory error rate as well, which is exactly what you would expect. But it's just another kind of cross-validation. So drug names with higher real-world error rates are significantly more likely to produce errors in the laboratory. So we see that this seems to be capturing some true underlying latent confusability of these names. This is just these two studies, again, to remind us about the relationship. And we see this as pretty strong cross-validation. The association is very similar looking, of a very similar magnitude. The fit of the models is a very similar magnitude. So we're pretty confident that whatever it is we're measuring, it's real. That is these kinds of psycholinguistic tests, which are very, very old. I used to have a mentor, a cognitive psychologist named Bill Brewer from the University of Illinois. When I started studying this stuff in the 1990s, he used to tease me. He said, "Are you still replicating Ebbinghaus?" And for those of you who know anything about the history of social science, Ebbinghaus was a German psychologist, one of the founders of experimental psychology, who in the 1800s was doing studies of word memory, not that dissimilar to what I'm doing in this study. So these are very, very old methods. There are literally thousands of word recognition, visual word recognition, auditory word recognition experiments, and word memory studies that go back to like the 1860s, 1870s, stuff like that. So I think this is fairly strong proof that laboratory-based measures of confusability are valid predictors, far from perfect, but valid predictors, especially on a relative basis. That is, the name that has a higher laboratory error rate is likely to have a higher error rate than the name that has the lower laboratory confusability. Comparison between the two studies, just a reminder that in 2017, we studied pairs. In 2025, we studied individual names. In 2017, there were 80 participants and only 37. And the later study was pharmacists and technicians only. In the first study, there were 170 pairs. In the second study, there were 77 individual names. In the first study, there were 4 tests. In the second study, we only took 3 of those. And it was pair-based rates in the first study, total error rates across all particular competitors. In the second study, very similar R-squared. We do think that the more recent study is a more accurate representation of what regulators have to do, because as I said before, regulators don't approve pairs of names. Regulators approve individual names. They're presented with an individual name and they have to say, yes, it's acceptable, or no, it's too confusing. So we think that's why we wanted to do this second study, to show this is how you would do stuff if it was an individual name. So, well, a couple of things. The model explains 30 to 35% of the variability in the real world error rate. Well, what about the other 65%? You could say, well, we didn't do a very good job. We only explained less than half of the variability in the underlying error rate. Well, there's lots of other things going on in real clinical practice, which we know contribute to wrong drug errors. And we didn't measure any of those things. So there's lookalike packaging. You can have 2 drugs from Lilly that both are, let's say, 100 milligram oral solid dosage forms. On a pharmacy shelf, they both have the Lilly trade dress. They have the Lilly brand identity. They look very, very similar, even if the names are completely dissimilar. So, and often if they're alphabetically adjacent, you have 2 Lilly 100 milligram oral solid dosage forms in bulk bottles on the pharmacy shelf adjacent to one another. This drives wrong drug errors in a way that has nothing to do with name similarity, other than maybe alphabetic adjacency. There's this other adjacent storage stuff. 2 names can be just next to each other because they're in the same class, because they're fast movers on a pharmacy shelf, etc. There's all sorts of computer system interface stuff, which we know causes problems. In the US, there are order sets that you can have a personalized order set. You can have names that you order frequently. These things can end up adjacent to one another on drop-down menus. And these interfaces, many of which are imperfect. So you can have autocomplete. We're trying to eradicate autocomplete in the US because of some very, very severe errors. There was this notorious error that happened to, I wrote an editorial about this nurse, Radon Vaught, in the United States, who mistakenly gave a patient vecuronium instead of Versed, and the patient died. It is a very celebrated case. The woman was brought up on criminal charges and actually found guilty, but then given probation. It's a very, very complicated case to go into. But one of the underlying causes of that is she entered VE into an automated dispensing cabinet. And this brought up vecuronium instead of Versed. And then there's all sorts of other reasons why she should have seen the difference but didn't, and so on. That's why she was convicted. But so autocomplete is very dangerous. This is just one example of a system interface that causes errors, especially if you allow for autocomplete of names that have similar beginnings. This would catch hydroxyzine and hydralazine as well. So there's environmental causes. The ISMP, the Institute for Safe Medication Practices, which many of you probably are familiar with, has some absolutely terrifying pictures. Although you who are the working clinicians on the call, you know that these terrifying conditions are just in your own hospitals and health systems. So the ISMP often will show pictures of a refrigerator where all sorts of refrigerated dosage forms are stored, just kind of, or drawers or shelves, which are chaotic, and where you have all sorts of small vials in a drawer or in a refrigerator or in a cart, which have identical trade dress, very small print. And again, these things can easily be confused because they're both in a similar sized vial with similar trade dress, even though they have nothing to do with name similarity. There's all sorts of other human factors: fatigue, workload, distraction, and data limitations. The self-reporting may not be, we may have predicted the real error rate, but self-report is not giving us the real error rate. There's actually measurement error in the outcome. So these are just a small list of all the things that we couldn't have measured in a study like ours. So it's a very, very noisy outcome. And given that amount of noise, it's remarkable that we were able to predict it to the extent that we were. And we think that's because the underlying factor, confusability, is such a powerful driver. And this is an upstream controllable factor. If you could use better pre-approval screening methods, then maybe a name like Durasol for a wart remover won't get on the market when there's an eye drop called Durazal already on the market. That's what we want to try to prevent. So implications for practice. For regulators, these screening tools exist. The evidence supports that they're valid. You could identify confusing names prior to approval. Health Canada has previously proposed to use similar tests, but the industry basically fought back and they didn't get used. There's some commercial brand screening companies that do this kind of stuff sort of in the background, but may not use the tests that I've recommended and validated. For manufacturers, you could test these names prior to submission. These tests are relatively inexpensive, especially compared to how much name is spent on branding or how expensive it is to change the name, like Brintellix had to do. And it can help you identify specific competitor names to avoid. And it's lower cost than recalls or patient harm. So what do I propose? I propose that you use this kind of standard validated 3-test battery as a standard component of pre-approval screening of drug names. And these findings validate screening accuracy. That is, they predict real-world error rates. They don't predict downstream harm. There's a bunch of other factors involved in harm. That is, how toxic are the underlying drugs? How badly do you need the drug that you missed instead of the drug that you got? How quickly do you recognize the adverse effects? How remediable are the adverse effects? So harm is more complicated than simply confusion. In fact, we know that the vast majority of medication errors in general have relatively little harm. And it's only a small proportion. So when people get lorazepam instead of clonazepam, there's essentially no harm. When people get cisplatin instead of carboplatin, there's fatal harm. So it really depends. When people get hydromorphone instead of morphine, there's very significant harm. But when people get two different antidepressants, there's often relatively little or no harm. So I wrote another editorial for the British Medical Journal relatively recently where I said, you're not going to fix this with one thing. Drug name confusion is a complex, situated problem that has to do with the names themselves and also has to do with the environment in which clinical medicine and pharmacy and nursing are practiced. So these are the other things that I think we should be doing to prevent wrong drug errors and name confusions. Starting with some of the strongest, which is barcode scanning. There's a great study, it's probably 10 years old by now. It was done at the Brigham and Women's Hospital and Mass General in Boston that showed a very, very dramatic reduction in wrong drug dispensing errors after the implementation of barcode scanning. So barcode scanning is just hugely effective. And in fact, the overall wrong drug error rate there's probably been a secular decline in these errors because of barcode scanning. Computerized physician order entry with alerts. We've shown in a couple of studies that if you give people indication alerts, that is, you give someone hydroxyzine and they don't have any allergy and they don't need any sleep medicine, but they have high blood pressure, they probably needed hydralazine, not hydroxyzine. So indication alerts can intercept wrong drug errors. The other kinds of CPOE alerts work. Tall man lettering. We haven't published this yet, and I've been a skeptic of tall man lettering for a long time, but we now have pretty strong evidence from Northwestern that tall man lettering dramatically reduces the rate of wrong drug errors in a large health system. You'll see those papers coming out in the next couple of years. I was surprised, but it seems to work. Separate storage areas. This is for high alert meds, especially when we know that two high alert drugs are confusing. You can change the labels. This has been done. Like cisplatin now has a colored lid and all these other things to prevent confusion, and pre-approval testing. So I've talked about some of the limitations: small sample size, US only. In the second study, only pharmacists and technicians, self-reported data, and so on. Future directions, like better validation, even better methods, and then implement this. We'd like regulators to actually demand that people use these pre-approval screening methods. It would add very little cost. So that's it. Lab tests of memory and perception significantly predict real-world name confusion errors, and we showed this in a couple of different studies. And thanks. And I'll now take questions. I'll stop sharing. I did stop sharing, I think. Yep. Okay, happy to take some questions now.
Thank you very much, Bruce. It was a fascinating talk, from the perspective of it being a widespread patient safety problem, but also in terms of the topic within that and the methodology used, I think was really interesting. So thank you very much. So if anyone's got any questions, they can put their hands up.
Yes, penicillin and penicillamine. People are talking about these other. The question, perhaps difficult to test, but is there any work to understand if the ease of pronunciation affects error rates, particularly when verbal checking? I have not looked at that. I have thought of that. Um, and I don't have that. I would suspect that ease of pronunciation, what it probably affects is short-term memory. First of all, there's the possibility of simply mispronouncing it and then someone hearing the wrong name because you said the wrong name. And we know, especially with like all these biologics, which are impossible to pronounce. And there's a perverse thing that's happened in the world of drug names. You might have observed the drug names keep getting weirder and weirder. Brand names keep getting weirder and weirder. So you have brand names like Skyrizi, which doesn't sound like any English word. Why do we have that? Because the drug companies are increasingly trying to find rare letter combinations that rarely occur in English and rarely occur in other drug names so as to maximize difference between existing drug names. So there's this perverse thing that's happening. In order to try to get distinctive drug names, we get drug names with weirder and weirder 2-letter and 3-letter combinations that rarely occur in English. So difficulty in pronounceability, especially of these long generic names, I think it affects short-term memory because short-term memory uses something called the phonological loop, which means when I'm trying to remember a phone number, let's say I'm trying to remember, 708 305 3383. The way I remember that is I silently to myself say 305 3383, 305 3383, 305. And any auditory interference disrupts this phonological loop. So if you can't pronounce the name, you can't rehearse it in the phonological loop very well, and you probably won't be able to remember it well. Second question. Is there a way, or have you used this to create something, an open source, to help them be able to try to test, to be able to try the test you designed for the lab to allow them to learn from your research about their... Oh, that's interesting. I haven't done that, but I'm an avid user of these AI coding apps now. I might be able to vibe code something like that, which is to quickly code up this sort of demonstration. Yeah, I mean, I taught in pharmacies at the University of Illinois College of Pharmacy for 22 years. Were students, did they know how susceptible they were to this? Probably not. But my impression from my former students was they were terrified of making errors. For all of you on the call who have been a young clinician or who train young clinicians, when they first go out and take care of real patients, they're terrified of hurting patients because of the enormous responsibility that falls on the shoulders of a practicing clinician because of the ever-present risk of really hurting somebody. I have an old pharmacy colleague. I used to ask him, well, he was a gerontology pharmacist, so we deal with these older patients on many drugs. And I said, well, how do you talk to your patients about adverse effects and medication harm? He says, I wait till they hurt a patient. And this doesn't mean he stands around and lets his students hurt patients. It means that inevitably his trainees will be involved in prescribing a medicine or dispensing a medicine or administering a medicine that harms a patient just because of the nature of medications. And then he uses that as the teachable moment. But that's a nice idea, Stephen. Adam asks, how do we bring industry into this with their corporate brand structures and dress and use the science to improve labeling and appearance, particularly with small dosage drugs like ampules where it's really, really hard, Adam. I don't think that the industry is completely opposed to this. I think if this could be kind of packaged up in a relatively easy test for them to do on a routine basis, they would probably do it. They do not ever want to change their names once it's out there. They'd have to retool all the labeling, all the marketing. It's a disaster for them when this happens. So they want to avoid it. So they pay trademark attorneys and stuff a lot of money to try to avoid this. I think with ISMP talks a lot about black print on an IV bag, which is transparent, where you can't read the print at all. Sometimes there's print on both sides of a transparent IV bag, and it's just impossible to read. Forget about the production pressure of a real clinician trying to read that quickly for a patient who's crashing and so on and so forth, or pulling an ampule out of a drawer or a crash cart. This is why these errors occur, because the actual environment where medicine gets practiced is so full of things that amplify the possibility for human error. So I think, Adam, rather than thinking the manufacturers are going to solve this problem with confusable ampules or small vials and stuff like that, I think we have to work much more to design the environment to make it more robust. Like I say, when I see pictures of these real world storage areas, either carts or drawers or refrigerators, it's terrifying because as a behavioral scientist, I know that refrigerator is absolutely a disaster waiting to happen. And even an automated dispensing machine, which were designed to prevent errors, if you allow auto completion, then you get errors like typing in VE and vecuronium comes up and the drawer opens for vecuronium instead of for Versed and the patient dies. So I would look for system errors to that, Adam, rather than thinking the manufacturers can solve. Some of these things we just can't fix just with more distinctive names. I think we need better order entry systems, lots of clinical decision support, and so on. Indication alerts. I think indication alerts are really, really useful for, especially at the order entry stage, because so many similar names have different indications. If your patient doesn't have the underlying indication, then it can alert you and say, oh, you've, prescribed metformin, but the patient doesn't have diabetes. Did you mean metronidazole? Something like that. Lisa asks, we need to be doing more to be telling patients that drugs aren't all wonder medicines and they need to be involved. Yes, absolutely. So there's a paper that I wrote probably 20 years ago now. The time flies, It's called Principles of Conservative Prescribing. And this is one of my foundational beliefs about patient safety, which is we should just be more careful. Drugs are incredibly dangerous, and there's a conspiracy of goodwill where clinicians want patients to feel better and to suffer less, and patients want to feel better and suffer less, and families want people, and drug companies want people to suffer less, and so on and so forth. So we think, well, let's just give them more treatment. But what we all know who study patient safety is more treatment just piles on the risk, and that drugs have tremendous risks and they cause tremendous harm. And if we just used fewer drugs at lower doses, so in our conservative prescribing paper, we're like, use non-drug therapy first. Don't use one drug to treat the side effects of another drug. Use older drugs. Use drugs at the lowest possible dose. There's all sorts of conservative practices which their side effect would be fewer medication errors. If patients are on fewer medications, there are fewer opportunities to give them the wrong drug, the wrong dose, the wrong route, lab pharmacy error, the contraindications, etc. So yes, simplification is an absolutely fundamental patient safety principle. It never goes away. It's always a good idea, and it cuts across so many kinds of harm. And you're right that we don't do it good enough because of this conspiracy of goodwill and because of the fear of, quote, scaring patients, which I've always thought was a bogus thing. I mean, it's not that patients won't get scared, but patients should be scared, right? I mean, we give patients these medicines. We give patients anticoagulants or opioids or, antidiabetics. They should be scared. Or antibiotics. These are dangerous drugs by any measure. They are objectively dangerous drugs. And in the US, there's a guy named Budnitz, Budnitz, who publishes, he's a federal government worker. He's published a series of studies now for like 20 years looking at emergency room visits and hospitalizations associated with adverse drug effects. And in the US, millions of people go to the emergency room and end up hospitalized because of adverse effects of medications taken as prescribed at the correct dose for the correct indication. This is not abuse of drugs. This is taking a drug as prescribed at the correct dose and ending up in the emergency room. So drugs are dangerous. And if we're going to scare patients about drugs, of course, we could comfort them and reassure them and tell them how to identify adverse effects, but we have to be able to be honest with them. These drugs are dangerous. They're likely to hurt you. We hope that the benefit exceeds the harm, and we wouldn't give them to you if we didn't think they did. But the possibility of harm is ever present. I don't see any more questions, but I'm happy to take more.
Thanks, Bruce. We have got a few more minutes, and if it's okay, I just wanted to ask a question if no one else has got any. But I'm a doctor and a prescriber, and over the last few years, we've had an electronic prescribing system introduced. And I'm just interested from your experience, how often these electronic prescribing systems are integrating the findings of research like yours, I guess, to improve safety and reduce risk. Because from my experience, I use them and think, I'm not sure they are. But...
So I think the answer is often. There is a cottage industry of academics like me, who do clinical informatics, try to test out our stuff in practice, and then try to implement it. Of course, we don't control. In the United States, there's essentially a monopoly. There's this there's a system called Epic, which has an overwhelming market dominance. It's used by most major commercial hospitals and health systems in the US. There's one other system called Cerner, which is in there's really two companies competing for all the business. And if it's not in Epic or Cerner, it's hard to get it in front of a lot of clinicians. So we have good evidence, for example, that these indication alerts. So an indication alert is for drugs that have a narrow set of indications. This doesn't work for all drugs. Like this wouldn't work for opioids or antibiotics, but for drugs with narrow indications. So metformin is a classic example. Metformin has a narrow set of indications. Basically diabetes and polycystic ovaries is what it's indicated for. If you order metformin and the problem list for your patient doesn't have diabetes or polycystic ovaries, we can just pop up an alert and say, Will, your patient doesn't have diabetes or polycystic ovaries. Are you sure you want to order metformin? Or would you like to add diabetes to the problem list? So it turns out this works extremely well. A lot of times diabetes just hasn't been added to the problem list yet. The patient really does have diabetes. And so you order the metformin and you add diabetes to the problem list. But a lot of the time in our studies, I think it was like 1% of the time, they canceled the order because they didn't mean to order metformin. They meant to order metronidazole or some other MET drug which they had incorrectly selected. So, and these things, another of my colleague, Jason Edelman at Columbia, he does this stuff about wrong patient errors, which are closely related to wrong drug errors. And he showed stuff like if you show the picture of a patient on the CPOE system when you're ordering, you can minimize the risk of wrong drug errors. Or with infants, who you can't tell the difference between the way they look because all infants or many infants look alike, that you could do things with naming of infants in the electronic medical record, which reduces the risk of wrong patient errors. So these innovations exist. Pushing them out to get them in Epic, for example, where they'll go across systems is much harder, but not impossible. So I think anyone on the call who's been practicing for a long time, you know that some of these things like barcoding, they do eventually reach the front line of practice and significantly improve care. My pleasure, Rebecca. Yeah, it was 6:30. I got up at 4:30 to make sure I had enough coffee to give the presentation. I think that's all the time we have, but if there's more questions, I'm happy to take them. You could easily reach me, bruce.lambert@northwestern.edu.
That's great. Yeah, thank you, Bruce. We'll probably have to get on to other meetings or clinics and things. So thank you very much for your time. And I'm sure people will be in touch if they've got other questions.
Bye, everybody. Keep up the good work. Thanks, Jenny.